So welcome to the Palaeontological Association Exceptional Lecture Series. This is a scheme that was started a few years ago in recognition of the fact that we have a wide body of excellent researchers in paleontology in UK, Europe, and beyond. And they are great vehicles with which to publicize the science of paleontology and reach new audiences, and especially non-specialist audiences that haven't particularly thought about paleontology before. Each year, we hold an application process whereby people can ask to be the exceptional lecturer. They will then receive money to visit various academic institutions who can apply to do so. Alex has been busy weaving his way across mostly Europe and the UK this year, visiting a variety of different audiences. You can host next year. If you were interested in hosting the exceptional lecturer, these applications are currently open until the end of the month to host Yara Haridi, talking about the evolution of bone. She's based at the University of Chicago, but we will try and support visits to as many places as we can within the budget that we have available. So do check your inbox for emails about Yara Haridi's talk and how you can apply to be a host. Moving ahead to this specific year, rather than looking forward, let's think about what's happening right now. And that's Alex, who is going to present the lecture from this year. So Alex is an esteemed researcher who has got a long history of research. He's been based at the University of Leeds for 11 years, and before that, studied at Bristol and Sheffield and a fellowship at Bath. His research interests are diverse, but they're united by big science questions about looking at macroevolutionary patterns, biases in the geological record, and mass extinctions, trying to make the connection between the biological and the geological processes. I mean, Alex, is that a fair way to sum it up? I'm putting words in your mouth there.
Yeah. More than fair, I think.
More than fair. Okay, so that's enough whispering from me. I think people haven't tuned in to listen to me. They've tuned in to listen to Alex. So I'm going to take a back seat and pass over to Alex to begin his lecture. We'll be sparing some time for some questions and answers at the end. So please let us know if you should be able to answer those questions anytime, but we'll answer them together at the end. Over to you, Alex.
Thank you very much, Rob. I'm just going to share my screen, so bear with me a minute while I get this working. Okay. Can we see that, Rob?
Yes, I currently see your presenter view. I don't know if you were able to maximize it so it's just the one slideshow view.
Sorry. It worked when we practiced.
We did practice this, I can assure everybody. We practiced that already.
Is that better?
Yes, thank you. That's looking great.
Marvelous. Fantastic. All right. Thanks very much. And thanks for joining us, guys. So I'm Alex Dunhill, and I'm going to be talking about species loss, community collapse, and ecosystem recovery during times of mass extinction. So first of all, big thanks to PalAss for giving me this award this year. It's allowed me to visit lots of different places across Europe, and also for the people who I didn't get to visit because I had lots of applications. I'm giving this online, recorded, live streamed, whatever you want to call it, lecture today. So Rob gave a really good introduction to what I do. But just to recap, I'm Alex Dunhill. I'm Associate Professor of Palaeobiology at the University of Leeds. My research focuses on macroevolution, paleoecology, biogeography, and particularly recently, all of those sort of topics have converged around sort of case studies involving mass extinction events. So I'm part of the Paleo at Leeds Research Cluster. It's a great place to do paleobiological research. And I have my little group within Paleo at Leeds called Deep Bio at Leeds, where we are mostly ecological modelers, sort of modeling ecological and evolutionary processes through Earth history. And these particular three, Amy, Lydia, and Annabelle, three of my postgraduate researchers, they're going to crop up throughout the talk today because I can't take credit for all this work. It's a team effort. And three of the main case studies I'll talk about today are the projects that have been led by these fantastic early career researchers. On top of that as well, it's a wider community effort. So a lot of the work I do is quite cross-disciplinary. I, by all means, don't have the expertise to do it all myself. So here's just a handful of people who will crop up throughout the talk. I won't name them all now, but I might name drop a few of them as we go through the talk today. So what's in the talk today? So first of all, what are mass extinctions and what do we know about them? And then I'm going to talk a little bit about what we don't know about mass extinctions. So some of the things that we're trying to find out within my group at Leeds, particularly by modeling community structure in the fossil record. And then I'll go through a few little case studies, some of the work that we've been doing at Leeds, before I'll talk a little bit about what we're going to get up to in the next few years. So first of all, what are mass extinction events? So mass extinctions are a sharp increase in the rate of extinction over a short period of geological time. And importantly, this raise in extinction rate results in a significant and prolonged reduction in global biodiversity. As paleobiologists, we usually define mass extinctions by extinction rates, which is the number of taxa going extinct per unit of time, usually per million years. And if we plot that through time from fossil data, it looks a little bit like this. When we have to compare our extinction rate that we get from the fossil record to background extinction. So extinction is a normal part of evolution. So there's an expected number of species that will go extinct for every unit of geological time. That's our background extinction. But as you'll note, that red wiggly line of extinction rates, there are some periods that really extend above that background rate that we would expect. And if we do what all sensible mass extinction paleobiologists do, remove the Cambrian because it's a bit weird, we then get 5 predominant peaks that correspond to the big 5 mass extinctions at the end of the Ordovician, the end of the Devonian, the end of the Permian, the end of the Triassic, and then at the end of the Cretaceous. But you'll also note there's some other intervals that stick up above that background extinction rate, so-called lesser mass extinction events. And actually thinking about the big 5 as just this isolated set of extinction rates is kind of outdated these days. As well as looking at extinction rates, which can be hard to sort of conceptualize as actual numbers, we can also measure mass extinctions by extinction magnitude. So this is the proportion of taxa going extinct across a point in geological time. Usually at the generic level, looking at marine invertebrates. And this gives us the figures that are traditionally used to define a mass extinction event. With the biggest being the late Permian, where we lose about 83% of marine invertebrate genera. And here's the big 5, the big pie charts, but we've got 2 of the more lesser extinction events for comparison here. And you can see that some of them are approaching that level of big 5 mass extinctions. So what do we know about mass extinctions? So we've got a pretty good handle of when they happened from looking at the fossil record, and also how bad they were. So we can calculate these extinction rates, these extinction magnitudes. We can look to other geological evidence, geochemical evidence, to get an idea about what caused them. We're pretty confident now in assigning causal mechanisms to most mass extinctions. Most of these correspond to sort of known Earth system perturbations, large igneous province volcanism, which causes all sorts of environmental cascades. Also big rocks from outer space every so often as well. And from looking at the fossil record, we can get a pretty good handle on what went extinct. So what sort of groups of organisms suffered most. And then we can study these organisms in detail, look at their ecology, ecological traits, and we can start to think about extinction selectivity. So start to create hypotheses as to why certain groups suffered more at certain extinction events than others. So we know quite a lot. I guess there's one other big question is, why do we care? These events happened a long, long time ago, not really relevant for the present day, perhaps. And I'm probably sort of preaching to the choir a little bit here in a virtual room full of paleobiologists, but I think it's always important to remember why paleobiology is not only interesting, but also important for sort of present day issues and society. So one thing that we know is happening today due to human pressures on the biosphere is that extinction rates are rising. But we don't really know how bad they're going to get, particularly when we're thinking about sort of climate change projections, because contemporary ecological records cannot inform us about extinction and biotic response to future warming predictions that we're likely to face over the next few centuries. However, the fossil record does, and in fact, it's the only direct evidence of extinction in response to extreme warming. And you can all include that in your grant applications if you want. So, what don't we know about mass extinctions, though? We don't know everything, obviously. There's always new stuff to find out. So the way we've thought about this is thinking about how extinction actually plays out at the community level. So we all know that evolution plays out at the population level. And if you put lots of populations together in one place, you form a community. So we're really interested in what role community structure plays in providing resilience to environmental change across mass extinction events. And that's simply because, myself included, the work I've done over the last 15 years or so, paleontologists almost always assume that extinction's a primary extinction event. So a primary extinction is when an organism goes extinct in direct response to an environmental pressure. So we can use a little schematic of a little community here of 4 different organisms. We've got a ray, a scallop, a lobster, and a clam. Maybe we've sampled these from the fossil record from one bed, and then we cross an extinction event, and in the next bed, the clam and the lobster have gone extinct. And as paleobiologists, we usually then look for some sort of selectivity pattern, some sort of set of ecological traits that might make these organisms vulnerable to whatever environmental change is happening across this event, with that assumption that they've gone extinct as primary extinctions, so in direct response to environmental change exceeding their ecological tolerances. However, we always have to bear in mind that within a community, they're not just single entities minding their own business, they're all interacting with each other. And one way to visualize this is in a food web, so we can put interactions, trophic interactions to link our taxa together. And then we can think about that scenario again. So maybe only the clam goes extinct as a primary extinction event, which then results in the loss of the lobster's food source. And maybe the lobster, maybe it's survived gasping a little bit, maybe, but it's survived that environmental change, but it's only gone extinct in a secondary extinction cascade because it has nothing left to eat. And if we think about mass extinctions where we're losing, say, 81%, 83% was it, of organisms within the ocean, ecological theory at this point suggests that many victims of mass extinctions would have perished because of these secondary extinction cascades rather than all going extinct as primary extinctions. And if we really want to understand how mass extinctions play out, we need to understand this, especially if we're going to use information from past mass extinctions to tell us anything about the biosphere today and into the future. So another thing we don't know that much about when it comes to mass extinctions is what happens afterwards in terms of recovery. So from existing studies of the fossil record, mass extinction rates of recovery seem to vary from less than 1 million to over 20 million years, depending on the event you're looking at, but also depending on how you measure and define ecosystem of recovery. And therefore, because there's lots of different ways to measure it, and no one really knows what's the best way to measure it, these estimates of recovery are disputed. So take the Permo-Triassic, for example, depending on how you measure ecosystem recovery, that change and that rate of recovery after the mass extinction can vary somewhat. And it's also, we don't really know whether ecosystem structure and function recovery recovers from mass extinctions in that it returns to that sort of pre-extinction level, or whether it returns to pre-extinction state or a new baseline. So have we got ecosystem restructuring rather than recovery? So these are some of the questions that we're interested in at Leeds. And in particular, we're interested in investigating that from a standpoint of investigating changes in community structure. So before we talk about that research, I think it's important for us to define what we mean by community structure. So biological communities are complex entities, and we can characterize these complex entities by their structure, which is the composition of the community. So thinking about the types of organisms that live there, the quantities of those organisms, and also importantly, how they interact with one another within that mix of populations. And one way to think about this is trophic structure. So the tiered organizations of organisms within an ecosystem that are arranged by predator-prey interactions. So in this sense, we're talking about food webs. So food webs being a number of food chains meshed together to form highly complex networks. And even though these are really complex, so they can be incredibly complicated, we can summarize and analyze them using a really well-defined set of key metrics that are known to reflect community structure and function. However, that's all well and good in the modern, when you can go out if you want to. It's not easy, but you can go out and observe organisms interacting with each other and feeding on one another. When it comes to the fossil record, we're dealing with long extinct communities, often of completely extinct organisms or even lineages. So that begs the question of what ate what, because it's something that we can actually never know for certain. So before I carry on, I think it's really important to show that I'm not the first person to have thought about these sort of questions in the fossil record. And there have been lots of analyses of trophic networks, of food webs in the fossil record. So if we go back sort of to the 90s to the early 2000s, these were usually just sort of visual depictions of really detailed expert studies of particular paleoecological communities, and that relied really heavily on the expertise of the people that studied these environments, these paleo environments and these paleo ecosystems for a long period of time. And there wasn't a massive amount of quantification added to these studies. They were just to demonstrate that sort of expert's view of what they thought was going on within that community. Fast forward 10, 20 years or so, and we have a few pioneers who've started to apply more quantitative community ecology methods to the fossil record. So Jennifer Dunn, in sort of the mid 2010s, started to apply some sort of rigorous quantitative metrics to fossil food webs to be able to compare their structure and function to modern day communities. And then also more recently, Peter Roopnarin's group working on sort of applying modeling, sophisticated modeling frameworks to looking at sort of community stability and resilience across mass extinction events. However, these studies still relied upon that expert opinion on those particular well-studied communities to be able to get a reasonable handle on what might have consumed what within those long extinct fossil communities. So as more of a meta-analytical paleobiologist, who was interested in these questions but didn't particularly have that level of expertise on one community, paleo community, never mind the sort of span of expertise that I'd required to do that for the sort of questions I wanted to ask. My aim was then to think about how we could go about modeling likely interactions between taxa based on their ecology that we can infer from the fossil record. So this project is what started all of this work. It was back in 2017, I think, when I worked with Andrew Beckerman at University of Sheffield, the chap in the hat. And then a little bit later on, Jack Shaw, who was then a PhD student at Yale University, on building a model that could reconstruct plausible feeding interactions amongst marine fossil organisms. So we looked at ecological trait data to inform the model, and we turned to this cube. This is the Bambach ecospace cube, which anyone who's done any marine functional ecology in the fossil record will no doubt be familiar with. It has three main traits, so tiering, feeding, and motility. And by combining these traits, you can define a really broad ecological niche or mode of life for that particular organism. And the more we thought about this model, it's actually also really informative of foraging theory, so how organisms find and consume their food. So first of all, there's the feeding trait, which basically, if you want to consume another organism, you have to be a predator. But then there's obviously lots of other feeding mechanisms within the marine environment as well. Tiering. So tiering is really informative of encounter rates, which is a really important part of foraging theory. It's basically, if you're going to eat something, you need to be encountering it within that same sort of spatial area. So tiering, where an organism lives, whether in the sediment, whether on the seabed, whether in the water column, is really informative for how the other organisms that particular organism is likely to encounter. Motility, so generally, if you want to catch something and consume it, you've got to be faster than it. You've got to be able to catch it. So in this case, the fast fish eats the slow fish. So the faster you are, the more likely you are to be able to consume other slower moving prey. The ecospace cube is missing one really important trait, though, particularly for foraging and feeding ecology, and that's body size. So basically, in the marine realm, body size is the main thing that dictates trophic relationships, because particularly for fish, if you're big enough to fit something in your mouth, you're going to have a good go at eating it. And I've been widely informed that bad things happen to animals that break that rule. And here's a picture to give that some evidence. So from these four traits, we built the Paleo Food Web Inference Model, or the FIM, silent P for short. It's a model written in R, and it's now published and freely available to play about with. So it works like this. You have an input, which is a taxon occurrence list, so a list of taxa that occur within your community, together with the four traits: fertility, feeding, motility, and body size. And then the model is basically just a set of inferential rules. So rules that dictate what can eat what based on each of these traits. I can show you how it works with an example of a tuna and a lobster. So can the lobster eat the tuna? So in trophic ecology, the arrow always denotes the flow of energy. So can the lobster eat the tuna? So let's look at the traits of these four critters. The tuna is pelagic, so swimming around in open water. The lobster is superficial. So this rule is violated because unless the lobster's got exceedingly long arms, he's not going to be able to reach up and catch the tuna that's swimming around in the water column. The tuna's very fast. The lobster's kind of slow, it's just crawling about on the seabed. So even if they were encountering each other in that same space, the lobster isn't going to be quick enough to catch that tuna. The tuna is very large. The lobster is pretty big, but it's nowhere near the size of a tuna. So even if it was in the same space, even if it was fast enough to catch the tuna, it isn't going to be able to subdue it to be able to consume it. And finally, they're both predators. So that rule is realized. So in this case, 3 out of 4 of the rules have been violated. You need to get 4 ticks across the board for a feasible feeding interaction to be realized within the model. One further observation here, the lobster seems to be further disadvantaged in this particular example because it appears to have already been cooked. So lobster cannot eat the tuna. So what the model does, it runs through every possible pairwise combination of predator and prey, and then it assigns these feasible feeding links that then basically denote the food web. And then the output is a visualization of the food web plus a whole host of structural metrics and functional motifs at the community and taxon level, which can tell us a whole lot about that structure and function of that community. So we're using this approach to answer, or to tackle, don't know if we'll answer them, tackle a number of big questions. So we're really interested in how extinction events play out at the community level. We're interested in how community structure then recovers after extinction events, and whether we get a recovery or do mass extinctions permanently restructure communities. So do we get that sort of shift of the baseline to a different ecological state? We're also interested in things like how communities respond to the loss of apex predators. So there's big predatory taxa that exert a lot of top-down control on our communities. What happens when we lose them? As they're some of our most threatened organisms in the marine realm today. So to show how far we've got with answering some of these questions, I'm going to take you on a catastrophic journey through the Phanerozoic. Bit of a lie, because we're going to fast forward through most of the Paleozoic, and we're going to start off at the Permo-Triassic boundary, in the earliest Mesozoic, actually, looking at the ecosystem recovery from the biggest mass extinction event of the Phanerozoic. Then we'll move on to the Jurassic, and we'll look at community collapse across the early Toarcian extinction, one of the lesser mass extinction events. Before zooming right to the end of the Mesozoic, we're going to look at community structural changes across the KPG mass extinction. And then almost up to the present day, we're going to look at the loss of apex predators across the Pliocene megafaunal extinction. So let's start off at the Permo-Triassic boundary, the end of the Paleozoic, start of the Mesozoic, where we're going to look at some of the work that Annabelle Nichols, current postgrad student in my lab, did as part of a research assistant role on a small NERC grant before starting her PhD. So what do we know about the Permo-Triassic mass extinction? Well, it happened 252 million years ago. We lose 83% of marine genera globally. We know from Rootnarine's cool modeling work that post-extinction community was likely unstable and therefore prone to further collapse. And this instability sort of continued throughout the lower Triassic, and we get a really long recovery interval, probably much longer than any other mass extinction event. What we don't really know is actually how long full ecological recovery took, and how that ecological recovery unfolded. So there's been a number of hypotheses thrown out there. So Chen and Benton provided a hypothetical model that suggested this was sort of step-by-step recovery of the trophic pyramid, with an aftermath, sort of classic disaster taxa aftermath of just a few primary producers and primary consumers, and they get that rebuild of trophic level by trophic level as you go through the different units of the early and middle Triassic. However, empirical evidence from the fossil record, at least in some places in the world, suggests this isn't the case, and that actually a mere million years after the mass extinction in South China, for example, here, we've got a fully recovered trophic pyramid, complete with big apex predator fish, these coelacanths, which shouldn't, according to the Chen and Benton model, happen for another 8 million years or so. So we wanted to test how marine communities recovered from the Permo-Triassic mass extinction by modeling marine food webs to construct a time series of fossil communities of interacting organisms throughout this recovery interval, and then quantify these changes using this approach to look at structure and function recovery to sort of determine the nature and timing of this ecological recovery from the biggest mass extinction event of the Phanerozoic. So our data for this comes from that paper I just talked about. We have the Guiyang biota just a million years after the mass extinction, then a whole series of other fossil Lagerstätten through the lower, middle, and upper Triassic. We turn to our two collaborators, Haijun Song and Paul Wignall, to give us that expertise, that subject level expertise on this time period and on these fossil communities. Annabelle coded up all of this data to feed into the FIM with help from Andrew and Jack. And then we built a series of communities through this interval. So here's our post-extinction recovery interval through the lower, middle, upper Triassic. And we have our 6 communities. You'll note that all of them look pretty well structured in terms of there's at least 4 trophic levels, 5 in some of them as we get later on. So there's no evidence really of that sort of step-by-step through the lower to higher trophic levels occurring through this interval. If we look at some of the metrics, we see a general trend of increasing alpha diversity within the communities, which sort of reaches a peak by the early middle Triassic. So these first 3 lower Triassic communities definitely have a lower diversity within the community than the later middle and upper Triassic communities. We see a general trend of increasing complex food web structure, but we see a big leap between these lower and middle Triassic communities. So the first 3 communities are not as complex as those later 3 in the middle and upper Triassic. So even though we're not seeing that sort of step-by-step, period-by-period rebuilding of the trophic pyramid, there's definitely a step in that recovery between our first 3 and our last 3 communities. And in particular, we see this through a general trend of increasing vertical complexity. So there's higher mean and max trophic level, a number of linear food chain motifs going up through our webs as we get into the middle and upper Triassic. So to summarize all that, the recovery of alpha diversity matches the recovery of gamma diversity, so global diversity, about 5 to 7 million years. And that also fits that recovery interval that was hypothesized by Chen and Benton. Their full recovery also takes a similar amount of time. But different to the Chen and Benton idea, those early Triassic communities retain 4 trophic levels and apex predators, but they are notably less complex than our middle to late Triassic communities. So there's definitely a leap in complexity as we move into the Anisian, into the middle Triassic epoch. It looks like ecological recovery takes longer than alpha diversity, but there's a big leap again between that early and middle Triassic. And our early Triassic communities, that even though they appear fairly complex, they're certainly not just really simple disaster taxa communities in this part of the world, but they appear a little bit top heavy. So there's a lot of diversity, ecological and taxonomic diversity at the higher trophic levels, and there's seemingly less going on in the lower trophic levels, particularly in the benthic realm. And this matches this global functional diversity signal, where we seem to get these weird reverse functional pyramids in that early Triassic interval with very rapid recovery in the pelagic realm, with lots of pelagic predators, fish, ammonoids, conodonts, and less slower recovery in the benthic realm, where we have most of our suspension and deposit feeders. So let's fast forward well into the Mesozoic now, or into the Jurassic, and we're going to look at community collapse across the early Toarcian extinction. So this was the first study that we applied these methods to, and it was originally funded by the PalAss via an undergrad bursary award. And I was working with Andrew at Sheffield on the food web model side, and we chose to apply it to a really well-studied data set of the early Toarcian extinction event on the Yorkshire coast. And I did this in collaboration with Chris Little, so expert on this particular event, who was also my colleague at Leeds. So what do we know about this event? 183 million years ago, we lose about 25% of marine genera globally, we think. So not as high as the big five mass extinctions. But locally, in certain parts of the world, particularly the Cleveland Basin, northeast England, because of really persistent dysoxia in these isolated marine basins, we see much higher extinction. So about 80% of marine benthic genera going extinct locally in some of these areas. And like the Permo-Triassic globally, in these really badly affected local areas, the recovery is really, really long. So locally in these Yorkshire sections, we see prolonged dysoxic conditions, and recovery is taking about at least 5 million years into the mid to late Toarcian. So what don't we know really? We don't really know how extinction dynamics play out within the community. We've only got that sort of before and after of before the extinction event when everything's fine, after the extinction event when a lot of things have gone. But we don't know how we get from that pre to post extinction in terms of primary extinctions and secondary extinctions. And then, like with that Permo-Triassic data set, we don't really know how ecological recovery unfolded as well. So all the data for this study comes from the Yorkshire coast. So this is Robin Hood's Bay. It's a beautiful part of the world. Also the best place in the world to get fish and chips. So if you've never been to Robin Hood's Bay, I recommend a visit on a nice summer day. So what were our objectives? So we wanted to test primary extinction trait selectivity. So what things were going extinct in response to environmental change. And then the species that were likely impacted by those primary extinction events, but then also the nature of any knock-on secondary extinction cascades that the loss of those organisms causes to the rest of the community. And then also we wanted to quantify changes in ecosystem structure and function to determine the nature of the recovery from this particular extinction event. So the data comes from years of study of this section, so dating back to Chris's PhD days, and subsequent sort of PhD students and postdocs who've worked on it since, including Jed Atkinson, pictured the guy in the hat and the nice moustache there. But also we did a bit of extra data collection as part of the PalAss funded undergrad bursary, which employed Carolina Zichney, who's now, I believe, just finishing her PhD at the University of Southampton. So together, all of that data was sort of meshed together to form 4 different intervals across the extinction event, which has been also published in a sort of a more traditional paleoecological sense in a paper led by Jed back in 2023. So Carolina coded up all of these occurrences. We fed them into an early version of the FIM, which has since been redone, and this paper was actually published last year, with help from Jack. And then we also used an R package called Cheddar to run some extinction cascade modeling, where we basically take our pre-extinction community from the fossil record and we subject that community to simulated extinction events. So we basically choose things to go extinct as primary extinctions and then see what happens to the rest of the community until it reaches either a state where nothing else goes extinct or the same diversity level as our post-extinction community. We could then do all sorts of complicated statistics that basically compare our simulated post-extinction with our empirical post-extinction to find our closest match, and therefore our most likely way that those communities collapsed. When it comes to primary extinction selectivity, you can do it randomly, so you can just make things go randomly extinct, or you can sort of make organisms more vulnerable to primary extinction based on their ecological trait. So you can select against certain ecological traits. And with that, we get this sort of framework of all these plausible extinction mechanisms that we can then compare by goodness of fit to our actual empirical post-extinction community. So if we look at what happens across this extinction event from our pre to post-extinction communities, we get a switch from a diverse community to a low diversity, high abundance community of opportunists. But as you can see, we don't see complete collapse of the community. But we see a number of telltale signs that this community is probably less robust as a result of losing a great deal of taxa from within it. We see increased connectance, so everything that's left is a little bit more dependent upon one another. Increased generality, so it means there's more generalists feeding across a greater proportion of prey that's left in the food web. And hand in hand with that comes increased vulnerability of the prey species. So that means that on average, the prey are being predated upon a proportionally higher level of predators. We see increased mean trophic level because the benthic realm gets hit harder than the pelagic realm. And we see the persistence of pelagic predators, but fewer sort of lower trophic level taxa surviving in the benthic realm. So we get these sort of top-heavy communities that we also saw in the aftermath of the Permo-Triassic. We see reduced competition and increased omnivory. So omnivory in this sense means feeding across different trophic levels. So the predators that are left are having to feed across different trophic levels to obtain enough food to survive. And if we look generally at these traits, we see a general loss of large-bodied, infaunal, motile, and predatory benthic taxa. So in terms of extinction dynamics, our simulations that are focusing on tiering, so primary extinction selectivity against particularly infaunal and benthic taxa, produce the best match to our post-extinction community. We were pleased with this, makes sense, because we're really confident that the main extinction driver within this section is bottom water dysoxia, bordering on anoxia at some point. So that would make sense that we're preferentially selecting against things that are living either in or on the seabed. And as a result of secondary extinction cascades within our models, it's the benthic predators that suffer the most secondary extinction events. In terms of recovery, so looking for our pre-extinction, post-extinction, early recovery, late recovery sections, we see a gradual recovery of diversity and structural recovery, which peaks in that late recovery interval. So at this point, and this really matches the sort of reoxygenation of the water column and the sediments. By the late recovery, we got returned to full oxic conditions. And actually, at this point, we've overshot the complexity of that pre-extinction community. We see a more complex community with a greater diversity of predators, greater amounts of competition, and increased specialization, which suggests that maybe we've got a restructuring going on here, and maybe that might be driven by sort of broader scale macroevolutionary patterns that are ongoing at this area in this interval, like the onset of the Mesozoic marine revolution, for example. So to summarize, primary extinction selectivity targets benthic, especially in faunal life habits. That matches with what we know about this extinction event and the extinction driver being bottom water dysoxia. Secondary extinctions are common amongst benthic predators. In particular, they're losing their prey. They also might be prime candidates for primary extinction as well, due to high energetic lifestyle with a declining oxygen content. The extinction event leads to top-heavy communities of pelagic generalists and also a few hardy benthic generalists that can eke out an existence in low oxygen conditions. So this is similar to the aftermath of the Permo-Triassic mass extinction. And also similar to that, particularly in this particular section, recovery is long and delayed by those low oxygen conditions. But that late recovery interval, when we've got the return to full oxygenation, we see more complexity. So maybe we've got also a restructuring of benthic communities due to the Mesozoic marine revolution. So let's zoom forward again through the Cretaceous, right to the end of the Cretaceous, in fact, and we're going to look at community structural changes across the Cretaceous-Palaeogene mass extinction event. So this is research carried out by Lydia Woods, who's a Leverhulme Extinction Studies DTP student. She's just writing it up now, so hopefully you'll see some of this stuff published in not too distant future. So what do we know about the KPG? Probably the most intensely studied mass extinction event. Happened 66 million years ago, about 40% marine genera extinct globally. Largely attributed to a extraterrestrial bolide impact, the Chicxulub meteorite. But we also have large igneous points volcanism in the Deccan Traps eruptions at or close to the KPG boundary, which has also often been linked to as a trigger for this mass extinction event. What don't we really know? So when it comes to the KPG, It's the most recent of the major mass extinction events to hit our planet. So it's been regarded as a candidate for the origins of modern marine community structure. So in the aftermath of the mass extinction, the recovery interval is when we get the origin of modern marine food webs and modern marine community structure. So that's something that we're interested in testing, is that did the KPG cause a permanent shift? And do our recovered Cenozoic communities look structurally different to our late Cretaceous communities? We're also interested in whether Deccan volcanism caused any structural changes prior to the KPG bolide impact. So there's always been these sort of hypotheses that maybe Deccan volcanism caused a sort of a weakening of the biosphere, which made that collapse after the meteorite impact more likely. So we want to test that as well. So our objectives were, did the KPG mass extinction cause permanent shifts in marine community structure? And did community structure change? It was a sort of a simplifying, a weakening of that community structure prior to the KPG boundary in response to Deccan volcanism. So the data that Lydia used for this study comes from a high latitude data set. So Seymour Island off the coast of the Antarctic Peninsula has probably the most extended KPG marine section in the world. So she utilized the expertise of two people who've actually been to Seymour Island and done a lot of fossil sampling out there in what sounds awful. It's just like an island of mud in freezing cold temperatures. So James Witts and Rowan Whittle were eternally grateful for their expertise and hard work gathering fossils and information about this event. So Lydia combined this with some data from the Palaeobiology database to come up with 4 really broad meta communities across the extinction window. She coded this up and fed it into the FIM with help from 2 of her supervisory team, Andrew and Jack. So this is what the data looks like. So we have a pre-Deccan interval that covers a little bit of the Campanian and most of the Maastrichtian. And then we have our late Maastrichtian interval, which is where the Deccan volcanism is ongoing within this interval. Then we have across the KPG into our recovery interval in the Danian. And then there's a gap in sedimentation, non-conformity, before we get a fully recovered community, which is of Eocene age. So across the KPG boundary itself, we see a diversity drop of 86%. So it's really high intensity, high magnitude extinction within this particular section. But I think probably the most surprising result we've ever got from any of these methodologies that was, despite this massive drop in diversity, our structural metrics remain fairly stable. So even though we've lost most things in most trophic levels, the structure is still pretty much the same. We do see some changes. So we see an increase in generalism. So there's more generalistic feeding habits. So all the specialists tend to go extinct. And we see a decrease in mean and max trophic level because we see most of the impact of the extinction event at the higher trophic levels, which is actually the complete opposite to what we saw at the Permo-Triassic and the Toarcian extinction. And we also see sort of less of these motifs. So these smaller scale 3-node interactions, which generally the more of these you find, the greater complexity you have within your community. So I think what we have here is actually a massive loss of functional redundancy. And this is a common pattern that we see in the fossil record in global data sets across mass extinction events, where you lose a hell of a lot of taxonomic diversity, but you don't lose a lot of broad functional diversity. And this idea has been named the skeleton crew hypothesis by Will Foster and colleagues, because in essence, you get that the ecosystem sort of limps on, the function and structure is retained, albeit by a skeleton crew of just a few taxa per broad ecological guild. And these sort of recovery skeleton crew communities are really vulnerable to further collapse if there's any further perturbation in that post-extinction interval. One important thing to note, once we get to the Eocene, our recovered community is almost identical in terms of its structure to our late Maastrichtian community. So there's no evidence of any permanent restructuring going on across the KPG mass extinction. It seems like we have this temporary perturbation caused by the mass extinction of the skeleton crew community, but then it recovers, even though we've got very different organisms inhabiting the recovered community. It's actually structured in a very similar way. So what about Deccan volcanism through the late Maastrichtian? So from that pre-Deccan to the late Maastrichtian interval, we see a diversity increase. Our broad structural metrics remain fairly stable. But if we look at these sort of more nuanced changes, we see a decrease in generalism, so an increase in specialism. We see an increase in mean and max trophic level, and we see more motifs. So there's actually an increase, a subtle one, but an increase in complexity through the interval when the Deccan traps are erupting. So there's no evidence at all that Deccan volcanism was causing any sort of negative effect on these marine communities in the lead up to the KPG mass extinction. So community structure is stable across the extinction event despite heavy species loss, and we have a maintenance of the function of the ecosystem by a skeleton crew. We do, however, see some changes. We see an increase in generalism and reduced vertical complexity. This is different in terms of this reduced vertical complexity to the Permo-Triassic and the Toarcian. So this is hinting at the importance maybe of bottom-up secondary extinction cascades in the KPG mass extinction case. This fits with what we know about this extinction event. Meteorite impact, primary productivity crisis will generate a lot of bottom-up cascades, which are going to preferentially impact the higher trophic levels within our communities. Once our community is recovered by the Eocene, we see no evidence of anything structurally different to what's happening in the late Cretaceous. So obviously we've got different things occupying each functional guild, but in terms of the broad community structure, the Eocene webs look very similar to the late Cretaceous one. So maybe the KPG is not the onset of modern marine community structure. And finally, Deccan volcanism did not drive negative changes in community structure, at least in this high latitude data set at Seymour Island. So let's fast forward before we get to the present day at the end of the talk. Let's fast forward to the Neogene. So now we're entering pretty recent time, about 2.5 million years ago, and we're going to look at the loss of apex predators across the Pliocene megafaunal extinction event. So this is work carried out by postgraduate student Amy Shipley. So Amy is a NERC Panorama student, also writing up at the moment, and you'll hopefully see this particular work that I'm going to present published within the next few months. If you're a PalAss regular, you'll be familiar, I think, with the figures on the next few slides, because Amy is a prolific PalAss annual meeting award winner, having won the President's Award for Best Talk and Poster Prize at the last two PalAss meetings. And you'll notice an upping of the in the game in terms of the graphics for this example compared to any of my graphics that I used earlier on in the talk. So what do we know about apex predator loss and the Pliocene megafaunal extinction? So we know that apex predators are important moderators of community structure, and they're currently some of the most threatened organisms in our oceans today. And we know that 2.5 million years ago or so, 36% of marine megafauna, including some apex predators, went extinct at the Pleistocene boundary. When it comes to the extinction of big vertebrate things in the ocean, through the Cenozoic, the Pliocene really stands out as having much higher extinction rates than any other epoch of the Cenozoic. If we look at what went extinct, a lot of marine mammals, particularly whales and big seals and things, also some birds and reptiles, but also some sharks as well. And one of those sharks that goes extinct in this event is the biggest predatory shark, and probably one of the biggest apex predators that's ever been on the planet, and that's Otodus megalodon. So the megalodon, megatoothed shark. So what don't we really know? So we don't really know how the global loss of an apex predator changes community structure, because even though apex predators are widely threatened and there have been some local extinctions, in the modern, we've yet to witness a major marine apex predator going extinct in recent time. So we're interested in what happens when that actually happens. So what happens when the megalodon went extinct? And did anything replace Otodus megalodon at the top of the Pleistocene food web? So Amy's objectives were to test whether the loss of an apex predator had a long-term or permanent effect on food web structure as we move from the Pliocene into the Pleistocene, and also what happened to that apex predator role occupied by the megalodon after its extinction. So Amy has quite a diverse team of supervisors. So there's Tracy Hayes, micropaleontologist, Jennifer Dunn, structural food web expert. On the data side, we have Catalina Pimiento, who's a world leading expert on this extinction event and on the megalodon shark. So she helped Amy devise a data set from the North Atlantic. It's by far the best sampled area for this interval, but also with some delving into the paleobiology database. And then on the method side, there's obviously me, but Andrew and Jack as well. So Amy coded up two really broad basin-wide meta-community assemblages for the Pliocene and the Pleistocene, fed them into the FIM to produce two really broad meta-communities. So we have our Pliocene web on the left and our Pleistocene meta-community web on the right. We have 6 trophic levels. So Megalodon is there, the big red shark sitting at the top in trophic level 6 in the Pliocene. And that matches a lot of sort of empirical evidence based on isotope analyses that suggest that the Megalodon's trophic level was much, much higher than any of our current top predators in the ocean, probably sitting up in that trophic level 6. We also have a sperm whale up in trophic level 6, which I won't talk about. But if you want to ask me about the sperm whale later, and there's time, I'll try and answer those questions. So across the extinction event, we lose our megalodon, and we lose a lot of other things, particularly from trophic levels 5 and 4. Despite this, our structural metrics are stable. Not really surprising, I guess, given what I just said about the KPG mass extinction. That common pattern we see across all of our extinction events. We do see an increase in generalism and a decrease in specialism. And like the KPG, across the Pliocene megafaunal event, we see that decrease in vertical complexity. And that's because we're losing things from the top of the food web. So there's fewer vertical food chains running up from bottom to top. So in terms of trophic role of apex predators, so here's what happens to each of them across the mass extinction event. So we see a general reduction in trophic level across the extinction event, and that's probably triggered by the loss of sort of baleen whales, and a lot of toothed whales and seals in trophic level 3, 4, and 5, which will then draw down the trophic level of those higher predators in the upper trophic level 5 and trophic level 6. The megalodon goes extinct, and also another large-bodied shark, Paratodus, in trophic level 5 goes extinct as well. The mechanism proposed for this extinction event has been cooling, sea level drop, and therefore the reduction in productive shelf area. So maybe we've got bottom-up cascades occurring, which are affecting a lot of these big baleen whales, making them go extinct, at which point the megalodon's losing its primary prey source, which maybe is bringing it into closer competition with the trophic level 5 predators. So thinking the modern day orca, the great white shark. So maybe some of this previous research suggesting that great white sharks killed off the megalodon via competition, maybe they're not so far off the mark, as nothing is replacing our big shark up in trophic level 6. So community structure is stable across the extinction event, but we see an increase in generalism and reduced vertical complexity. So similar to the KPG mass extinction, bottom-up cascades probably happening. Nothing replaces our megatooth shark as a super apex predator, but we see a reduced trophic level of all top predators across the event. Maybe that's triggering this increased competition between trophic level 5 and 6 after the loss of a lot of these large-bodied prey that megalodon was supposedly mostly feeding upon. I think it's worth noting out of all the studies we've looked at, our structural food web approach is probably falling a little bit short in answering some questions with this particular data set, because our bottom sort of our top-down control of our apex predators probably relies a lot upon sort of population sizes and being able to quantify that, which we're currently unable to do with these structural food webs. So with that in mind, what's next? So the stuff that we're currently working on at the moment, we're really still interested in when did modern marine ecosystem structure originate? We've not really answered that. We've had some hints that maybe we've picked up some sort of signals of the Mesozoic marine revolution in that early Jurassic data set. And then maybe by the KPG, we've got a modern looking community, even before the KPG mass extinction in the late Cretaceous. So Annabelle's current PhD project is looking at that intervening period in between, so in the Jurassic through into the Cretaceous, looking at community structural complexity changes to try and pin down if at any point or a sort of a series of events that might have led to modern marine ecosystem structure through that interval that we associate with the Mesozoic marine revolution. We're also interested in why there hasn't been a major mass extinction in the last 66 million years. So we have a current NERC NSF grant called Cascade, which has employed Baran Karapanar as a postdoc with me in Leeds. So some of the things he's looking at is how ecosystems collapsed across different Paleozoic and Mesozoic and Cenozoic environmental perturbations, and whether actually we're seeing an increase in the robustness of our communities through the Mesozoic and Cenozoic compared to our Paleozoic communities, and maybe that's why we're seeing less frequent mass extinction as we head through the Mesozoic and Cenozoic. However, a lot of that depends on what happens at the base of our food webs. So the eagle-eyed amongst you might have noticed that the base of our food web is a single node for primary producers. But if we want to be able to say anything about these sort of bottom-up cascades, we really need to be able to quantify what's going on at the base of the food web. So together with some Earth system modeling that Baran's getting to grips with, we have another postdoc at Sheffield, so Tanya Stridham, who's going to be applying our approach, but in a dynamic food web sort of scenario where we can actually play about with energy flow and population sizes. And finally, I guess our main aim is to try and help us understand how modern marine systems might respond to a mass extinction event. So what would happen if we subjected a modern marine community to, say, a Permo-Triassic style mass extinction? Would we see the same type of collapse that the Earth witnessed 252 million years ago? Or more positively, are our modern communities more resilient to those sort of changes and secondary extinction cascades than our more ancient Paleozoic ecosystems were? So this is the Cascade team. So we're going to be trying to answer some of these questions across the next 2 or 3 years. And I'll stop there. I think I've been talking for about 50 minutes. That's long enough to listen to me. And I can hand back to Rob, who can moderate any questions.
Thank you, Alex. I'm going to give you a round of applause from just me, but we will imagine that everyone's clapping along at home when we can't hear them. Without
I'd like to think so.
There was an awful lot of food for thought in there. I was busy writing down lots and lots of questions, but I think probably should put a pin in my questions and start turning to the audience instead. So I've asked people to post their questions as they see fit in the little Q&A box. So some have come through already. I might try and allow time for one of my own questions at least, but let's start with some other people's questions first, because I should prioritize them. Okay, so Megan asks, are there plans to develop the food web model to include increased complexity? So she raises the example of scavenging, and I was thinking about that as you were talking, in the sense that if I think about whale fall or jellyfish, these are major components of ecosystems, but a little tiny annelid worm is busy eating a whale, but in your model that wouldn't count. Is scavenging very different from predation in this sense?
So scavengers are difficult to anything but predator-prey interaction is difficult to quantify within the structural frameworks that I've talked about today, right? Because essentially what you have is just a directional predator-prey interaction. And Baran has played about with putting scavengers in, but what you end up with are sort of trophic level 7 parasitic, or parasites as well. Parasites are even harder. You end up with sort of trophic level 7 parasitic snails or scavenging worms, which is completely unrealistic for our a simple structural framework. But one thing we're looking at doing, because we've got this sort of Earth system modeling framework that we're applying to our dynamic models, is that we're wanting to also try and experiment with working in sort of nutrient feedback loops, which wouldn't necessarily mean we have to include individual scavengers into the network. But what we're trying to do is balance sort of energy flow through the system.
Lovely.
Which will allow that sort of nutrient flow, particularly from pelagic to benthic systems, right? Which is basically how, particularly our deep benthic systems, how they sustain themselves, right? So good question, and it's something we're experimenting with, but it's tricky.
Thanks, Alex. For the question and answer session, perhaps I should suggest that you maybe stop screen sharing. I mean, it's possible we need to ask you to come back to have a look at it, but right now, maybe people want to see your lovely face. Thank you, Alex.
Yeah.
Okay, so Andrea asked a question. Why would you think structural metrics were kept stable after the KPG mass extinction? And would you say it was due to luck or something else? What would have happened if the structure wouldn't have been the same, like if some species were to have gone extinct? Would it be possible for another type of structural metric to have occurred?
That's a good question. So I think the thing that's happened within our, and a lot of this is probably a result of our course methodology as well, right? So our structural webs are really conservative because they essentially are just presence absence of taxa or guilds sometimes, and then the feeding interactions that connect them, right? You can only have a taxon there or not. Whereas in reality, across these extinction events, you would have got population crashes, wildly fluctuating populations across different parts of the community. You might have had a few generalists that absolutely dominate and then a few rare taxa that just cling on. So we can't get at all those different sort of nuanced sort of conservative structural approach. What's happened as far as we can see within our modeling frameworks is that you're getting a few species surviving within each broad structural guild, right? And that maintains that structure. So in essence, you've got the same structure post-extinction, even though you've got fewer taxa left, you've just got massively reduced functional redundancy. So in the pre-extinction community, you've got a lot of things doing similar things to each other. And that's why you get that raised level of competition and all those other increased complexity, signals of increased complexity, right? But in the aftermath, most of those things within each guild have gone extinct. And you could hypothesize, which I think this has been thrown out there in sort of the functional paleoecology literature, that this is probably due to as things get tough, as environmental pressures increase across the extinction event, those organisms that are occupying sort of broad niche, the same broad niche, can't maintain that niche separation anymore. So they're drawn into tighter competition for dwindling resources or whatever. And you get heightened levels of extinction within those clusters of functionally redundant taxa. So only a few within each guild survive. But that structure is essentially at the broadest level the same pretty much as that pre-extinction structure. I suspect if we can get the dynamic modeling working and making sense, I suspect we'll see much more pronounced changes once we can account for changes in energy, changes in biomass, changes in population sizes.
Well, thanks, Alex. I guess that immediately makes me start thinking about abundance, but abundance is going to be locked up with all sorts of really weird issues that you might be able to do in a modern ecosystem, but a fossil ecosystem is going to be very hard. But we'll come back to that again. So I'm sorry to everybody. There's more questions that we can possibly get through, so I won't be able to ask everybody's questions. Apologies, because we need to try and keep this manageable. We can't go till midnight. But there's a question from Emma. Fabulous talk. A cheeky must ask. How sensitive is the approach with the PFIM? So can you remind me what the PFIM stands for?
Paleo Food Web Inference Model.
Paleo Food Web Inference Model. How sensitive is the approach to incomplete fossil data? Can it deal with data/sampling gaps, or do you find that the results coming from it aren't sensitive to gaps?
Very good question. So all the examples I presented today, we haven't corrected for any sort of sampling biases. One, because it's really difficult, because essentially we're trying to quantify what's not there, which is quite hard. But we have, well, I say we, so Jack, as part of his PhD, Jack Shaw, did test how well, first of all, the FIM reproduces well-known, well-constrained modern marine food webs, does pretty well. It does as well as most other modeling frameworks that are used in modern ecology. Because actually, so food web models, interaction modeling is well used in modern ecology as well, particularly in marine ecology. It's really difficult to watch things eat things in the sea, basically. But there's a lot of sort of ecological data available for modern taxa that's used to parameterize these models that we can't get at in the fossil record. But the FIM performs pretty well. What it does really well is it reconstructs known feeding interactions in modern data sets really well. But it also has a higher error rate when it comes to it reconstructs feeding interactions that aren't realized as well. And I think that's part of we've been grappling with that. I think that's a different issue to what the question was asked, but we've been grappling with that a little bit as to why that might be, which I can talk about if you want me to, or I can just gloss over to the sampling bias thing. But Jack's also applied it to sort of modern ecosystems that he's then sort of put through fossilization filters. So thinking about the hard-bodied things that are likely to preserve and then removing the soft-bodied things that are really unlikely to preserve. Broad level structure is not massively affected by losing squishy things. But there are telltale signs of losing squishy things that happen in certain parts of the web. So most of our squishy things are low trophic level. They are sort of deposit feeders and suspension feeders. So removing a higher proportion from that part of the web does cause notable changes. So generally you'll overestimate generality, you'll underestimate vulnerability, and you will underestimate mean trophic level as well. So it's predictable. So one thing you can, as long as you're confident that your preservation regime isn't changing across the interval you're looking at, you shouldn't be creating any artificial impacts across the extinction. Obviously, if your environment drastically changes, your preservation regime drastically changes, that could be problematic. You might just be reporting an artifactual
I'm going to
change.
take chair's privilege here and ask a couple of follow-up questions for Emma. But I guess because that was also something I was thinking about was the soft-bodied things. If I think about an ecosystem, the algae and the jellyfish in a marine ecosystem are going to be mega components, which you ordinarily can't get a hold of. But I guess that's what you were talking about, right? About the resilience of the sampling method to survive those sorts of soft, hard fossilization biases. I guess I was just thinking about the abundance of the jellyfish and the abundance of the algae is enormous. But a different question that's slightly related to Emma's point and what you were just saying there was about fossilization biases even within the hard body things. I mean, when you're looking at the mega scale that you are, like 20, 30 million years, you're almost certainly going to be picking up changes in sedimentation rates and dissolution and big mega taphonomic biases that might potentially distort the lens with which you're looking at the fossil record. I mean, I shouldn't. Look at who I'm asking this question of. I'm asking someone that's written papers all about that very topic. But are you able to account for that, or take it away, or is it not necessary? Or
So I think in the data sets we've presented, I'm not massively concerned because I mean the Permo-Triassic one, we're looking at a series of snapshots of Lagerstätten, and there will be differences, right? But generally the preservation is exceptional in all of those data sets. Across the Toarcian, we don't see a massive change. There's a slight deepening and shallowing, and particularly that early recovery interval is supposedly less well sampled. That's why we targeted that with a bit of extra sampling. But the biasing would have to be quite extreme, I think, to really change our result because we're not losing taxa. But there may be that recovery happened a bit sooner, I guess, than right in that late recovery interval. But it matches with the sedimentary record as well. We've still got low oxygen conditions where we'd expect to see better preservation, right, in that early recovery interval. And then same for the KPG. The Seymour Island is a really good case study because the preservation it doesn't really change across the extinction boundary, which is quite often across mass. That's one of the problems with mass extinctions is because you've got such environmental upheaval, you often see big sea level changes and things like that, right? Across the KPG, you don't really get that across through Seymour Island, so we're fairly confident. But it is something we're looking at. We're looking at how we can start to account for sampling biases, because where we want to take this next is we want to look across longer timescales. We want to look across Phanerozoic timescales, and we want to compare how community structure changes across the Phanerozoic. And at that point, we really do need to think about all these sort of ways of controlling for the stuff that's missing. And I don't have an answer as to how we're going to do that yet.
I realize that we're running out of time, so I probably, although time does go on and on, I don't want to take too long. Some of the people's questions that have been asked have come up in conversation in the sense that we've just talked about them. One of them was about the primary producer singularity data point. We've talked about that in the answers. One of the answers about fossil preservation biases, but we've just talked about that. So I think some of those questions have come up. So maybe I'll just be extra greedy and save that last question for me, because I'm an egomaniac and I want to know about this, which is the Devonian. So you've focused very heavily on the Mesozoic. I'm a big Devonian fan. I mean, I guess that's partly because I've worked on the Devonian and that's what I'm interested in. Well, alongside many other things. But the Devonian, it sounds like it would be a really interesting place for this to be looked at, because this is a construction of different food webs. I mean, rather than thinking about destruction, we're seeing construction, in the sense that we're seeing tearing, invasion of the pelagic realm, and it concludes with this mass extinction event, or series of pulses of mass extinction events. So I'm particularly interested in the Devonian. Please, will you roll this out to the Devonian? Or is that's not really a question, that's more of a beg.
Yeah, I mean, we have a I say we, it's that's more Paul Wignall's domain than mine, the Devonian. But I mean, I am sort of helping out with a couple of his projects at the moment. They're not necessarily food webby at the moment, but they could be. But yeah, if anyone has good Devonian data set, including you, Rob.
We did try and construct the interactions of some of these things, but that was quite hard work on the basis of just looking at one proof of taxa. So automating it for hundreds, I don't know how you do it.
Well, I'm open to ideas.
Lovely. Well, this is me now trying to invite myself into a research project, which is not what this is about. Thank you, everybody, for. I realized I didn't introduce myself at all at any point, but that's because this isn't about me. I'm an elected member of the PalAss Council and trustee as the meetings coordinator, Robert Sansa. So I'm introducing Alex on behalf of the Palaeontological Association. So just as a quick couple of reminders, please do, if you want to host an excellent talk like Alex's, well, not Alex's, it's now been and gone, but we're looking forward to Yara Haridi for the next one, please do take a look at the PalAss website, the Palaeontological Association website, with information about how you can act as a host, as various different people did for Alex in Prague, in Oslo, Warsaw. Where else have you been, Alex?
Oxford, London, Glasgow.
So Alex has been a busy bee. You could be one of those destinations for the next visit. And at that point, I should thank you, Alex, for your time and the excellent research from you and everybody on your team. So it was a pleasure.
Thanks, Rob. It's been a pleasure. Yeah, thanks, PalAss.
Thank you.